Hi everybody, and welcome to Insilico Cambridge. We haven't officially opened yet, but we're in the process of moving. My name is Alex Javankov, and I'm the founder and co-CEO of Insilico. So today, let me welcome you to our site. So as you can see, it's a pretty epic address, 1000 Mass Avenue. And let me just show you where we are. Which is a really cool location. So 1000 Mass Avenue, it's right next to Harvard, and you can actually have a look at the map here. Soon we're going to be open for visitors. And this product launch, we are going to do in a very unconventional style. Hey guys. Very unconventional style where we learned a little bit from Neuralink and got inspired by how Elon did this for the update. Of course, what we are doing here is not as, may not seem as cool as what he's managed to do with Neuralink, but we're going to show product demos. And instead of just showing PowerPoint slides, we want to figure out how to show you the actual benefits of the product. And here we have Dr. Petrina Kamia. She flew in from Montreal, Canada for a conference here, but she is going to be presenting some of our tools today. We're going to have a full house of Insilico executives responsible for the platform technology. It's going to be myself, Petrina, Dr. Alex Alaper, and Dr. Khadija Alavi, who is going to present Precious 3 GPT. We're going to present multiple tools, and we're going to also explain our new sustainability initiative. And Petrina, while I'm setting up, can you tell us a little bit about your history at Insilico and how did we come to sustainability? Because we've been doing that right when you joined.
Right. So I joined Insilico Medicine in August of 2020. Back then, we had a platform, an end-to-end platform for drug discovery and development called Pharma.AI. But at that same year, we also partnered with Syngenta on the development of sustainable herbicides and pesticides. I do believe that it was 2 years later that we began to work with Aramco on the development of carbon capture materials. These two initiatives, sustainability initiatives, have led us to grow further in this space to further build on our vision, which is to bring healthy longevity to everyone on the planet. Without sustainability, there is no healthy longevity. So that's basically it.
Well, thank you. And I also believe that we shouldn't excuse ourselves for expanding into sustainability as we have to do that in front of investors sometime. We actually are making nice revenue here. And let me switch to the screen now, and you would be able to see what I'm doing. So let's go to insilico.com, and I'm going to show you IMGAIA initiative, which we are going to show today. So today what we're going to present to you is our sustainability initiative. It's going to be presented by Alex Alaper. Right after my introduction, we're going to talk about our initiatives in CO2 capture, base oils, hydrogen storage, lubricants, and agrochemistry. And we're going to also do a massive new product release presented by myself and Dr. Petrina Kamia and Dr. Khadija Alavi. So we're going to show you really cool tools. DORA is our draft outline research assistant. Really cool tool which allows you to write research papers from scratch and create all kinds of scientific documents and perform multiple experiments at the same time. we're going to present PandaOmics Box. I'm going to present that. That's our very new hardware solution for hospitals and for biotechnology companies who do not want their data to be passed onto the cloud. We want to make sure that the data is 100% secure, and we developed a solution which allows you to do everything on premise. Even the on-device compute is done via on-chip confidential computing. The box doesn't have Wi-Fi, and it has many capabilities of our PandaOmics platform and DORA. We're also going to show Precious 3GPT. That is something that I'm extremely passionate about. You know that everything I care about is about aging research. So we use aging research and AI as a platform for drug discovery, and now have multiple human clinical trials running from zero with very highly promising targets with massive potential for indication expansion. And this tool is a multimodal, multi-omics, multi-species transformer where we tokenize multiple data types separately and train one global model to perform multiple tasks. I'm not going to steal the thunder, but I'm extremely excited about this one. And finally, we're going to present an Insilico design kit. So whenever you want to write a cool research paper or a PowerPoint presentation about AI, you might want to utilize a standard set of icons, and we provide that. I'm going to show it to you as well. So let's start with DORA. DORA is going to be presented by Dr. Petrina Kamia. I just want to give you a little bit of a gist. Let's log in. And this tool allows you to choose from multiple templates. The template you like, so for example, for doctors, you can use case reports, right? So research paper describing a rare case report where you want to present to your management or turn it into a scientific paper. Conference abstract, scientific blog post, grant application, review paper, original research article, and there are going to be many more original research articles presented. So for example, in this case, you can implicate a gene in a specific disease and give some hypothesis. So for example, of course, my favorite one is, let's say, tinic. And the disease would be, let's say, cancer. And my context would be tinic is implicated in aging and age-related diseases. What is a potent modulator or potent driver of multiple cancers. And that's my hypothesis. The system allows you to choose from a variety of sections that you would use in a scientific publication. You can generate the document overview. It started working, and in about a few minutes, it will give me a complete article. I would be able to modify it, position the references, and see if I like it, and I would be able to modify it. So you can see that it generated a bunch of suggestions and a discussion, and now we will generate the content. So now it's running, and you may wait up to 30 minutes for the document to be created. Let's see if we can do it in a shorter period of time. And here you can actually see the progress as sections get generated. You will be able to edit them right away. So this system utilizes multiple large language models working with specific domain-specific tools that perform original research. It utilizes an army of agents that were specifically designed to perform original research, because we want to eventually get to breakthroughs that were impossible to achieve utilizing human intelligence or even large working teams. So we want to demonstrate that it is possible already today. With time, this system is going to improve dramatically and allow you to perform original research with a specific objective. We hope to deliver it within this year. And let me go back to IMGAIA. And show you another really cool tool, which is our design kit. So if you go to our design kit and download the Insilico design kit, it is a PowerPoint presentation with many, many, many different icons. So accept all, let me get that. In this particular presentation, you have, of course, usage license. It's open access, open source for non-commercial use. You can utilize multiple different icons and workflows, going from computers to business documents to life sustainability, new materials, in vivo experiments. You can go and select any one of them. If you like to describe agents, describe very specific complicated scenarios and workflows for your scientific publications, you would be able to use many of those icons in a standardized format. You will also be able to see some examples, if you like timelines, all kinds of transformer architectures, all kinds of scientific workflows and Gantt charts, specific formats for generative modeling, presentations of your research papers, presentations of different verticals. You would be able to present the various ways your company is organized or a specific workflow. By the way, here you can see the way Insilico is organized. So we have an Insilico commander running an army of agents, operating a model orchestra of more than 700 domain-specific validated models with experimental validation. We have biology tools, biologics tools, small molecules, drugs in clinical trials. We have a large community of software users, so please use DORA and PandaOmics more. We have competence in quantum computing. It's still being, we're still building this expertise. We have robotics, also run by agents. We work with aging research, and now we're also adding the sustainability angle with sustainable agriculture, CO2 capture and recycling, new materials, new energy chemistry, and lubricants. And we're adding many, many more additional verticals as we go, because we are primarily the AI company, even though we've managed to learn how to produce really ultra high quality drugs that people are willing to pay billions of dollars for with very substantial upfronts as they progress into human clinical trials. So we also want to help the environment. Now I'm going to pass the word to Alex Alper, our president. He is going to dial in from the UAE, from Abu Dhabi. We have a pretty substantial site in the Middle East, and we are actually very bullish on that region because it's one of those regions that can now, utilizing our resources, can convert petrodollars into life-saving therapeutics, and we want to enable local countries to do that at scale, and also support them with sustainability initiatives. So let's check if DORA produced something valuable in terms of research paper results. So you can see that it actually came up with a really nice title: Dual Role of TNIC in Aging and Cancer, Consequences for Therapeutic Targeting. So I would probably use something like implications, or so you can very easily edit it right from the console you can see it produced an abstract, and it already included the references. So you can go and see those references. In the references section, you can see citations, the year it was published, and the journal, in addition to PMID. The references are pretty good, so we've trained the system to produce very actionable references. And you can also see that it used a bunch of tools inside PandaOmics. So it referenced PandaOmics as one of the key tools that were used for research. And if you want to go specifically into the hypothesis generation methodology, the system utilizes PandaOmics in order to generate a knowledge graph and then explores the knowledge graph. So you can get a subscription to PandaOmics and derive those beautiful pictures that you can enrich your paper with if you are publishing a paper. And also it gives you future research directions. If you want to do experimental validation, you can try it out in different models. I see that it even suggests some inhibitors, not ours, that were previously known, and I know that one very well. So yeah, you can utilize this at scale, and if you like, you can export it. You can export it in Word or in PDF. So the document has been successfully exported, boom, and you can go and edit it offline. So that's DORA. It's integrated in multiple tools that Insilico is supporting, mainly PandaOmics, and you can utilize it already today. It will get better with time. We have the development plan all the way to December this year. The teams are fully occupied working on this tool, and we really hope to enable more people to get into science. Thank you very much, and please enjoy the show. I'll be with you one more time for the PandaOmics Box reveal.
At Insilico Medicine, we believe in a future where science and sustainability go hand in hand, paving the way for a healthier planet and healthier lives. In 2023, Insilico Medicine established its largest regional site, in the International Renewable Energy Agency headquarters building, in Masdar City, UAE, which hosts innovative agencies and companies working on sustainable solutions.
At Insilico Medicine, our mission is to extend healthy, productive longevity. This mission isn't just about individual health, it's about the health of our planet. We understand that sustainable world is essential for long-term human well-being. Today, we're announcing the launch of Generative AI for Environmental Sustainability Consortium. This consortium will have 4 major focus areas: sustainable gas capture, including carbon capture and hydrogen storage, agrochemistry, and base oils and lubricants. Why these areas are important? CO2 capture is a collection of technologies that minimize CO2 emissions generated from man-made sources such as burning fossil fuels before it is released in the atmosphere. Scientists are also considering direct air capture, where CO2 can be removed from the air using a series of chemical and physical processes. Hydrogen is an ideal clean energy source because its sole product after combustion is water, and no greenhouse gases and environmentally harmful compounds are released. Hydrogen is of particular interest in automotive applications because of its zero-emission fuel, abundantly available, and when liquefied, has triple the energy of gasoline. Full system based on hydrogen involves its production, delivery, and storage, as well as fuel cell technology. Among them, hydrogen storage plays a key role in this development. Sustainable agrochemistry integrates principles of organic chemistry, biochemistry, and environmental science to develop innovative solutions for the agricultural industry. By advancing our understanding of how chemical inputs affect agricultural system, agrochemistry plays a crucial role in meeting the growing demand for food in a sustainable and environmentally responsible manner. And finally, lubricants are substances that are applied to surfaces in mutual contact to minimize friction and wear between them. So improved lubrication leads to a smoother operation, lower maintenance needs, and better overall performance of machines and systems. We invite both academic and industry institutions and key opinion leaders to join the Sustainability Consortium. We're happy to share that we've already obtained commitments and interest from several players, including such prominent AI experts as Alan Spurgudzik Group from University of Toronto and the Acceleration Consortium. Our goal of the consortium is to enable scientists with a platform and the framework to benchmark their AI models, exchange data and ideas. We designed the platform to be fully open source so everyone can freely use it to advance the field without any restrictions. If you're interested in joining the consortium, please visit Insilico's website Sustainability Consortium page or send an email to sustainability@insilico.com. We're looking forward to working closely with you and advance the field together.
Thank you, Alex. Good afternoon. My name is Patrina, and I'm the president of Insilico Medicine Canada. I'm also the global head of AI platforms at Insilico Medicine, and today I'm proud to introduce you to Science 42 DORA, the latest member of the Pharma.AI family of applications. Science 42 DORA is our first AI-powered draft outline research assistant. Using the power of AI, DORA seamlessly and autonomously generates drafts of scientific documents based on the end user's specified templates. Users can edit the generated text with DORA's intuitive, user-friendly interface and leverage functions like AI-powered tuning and on-demand searches for relevant references. Worried about DORA hallucinating? For all the content generated by DORA, a list of relevant references for every topic is provided as legitimate evidence. Let's watch DORA in action.
Hello, my name is Julia Smolik, and I am a bioinformatician at Insilico Medicine. The rapid increase in scientific research across diverse disciplines has led to an overwhelming volume of published studies. Review papers have therefore become indispensable, as they not only organize existing analysis, but also critically assess current knowledge, identify research gaps, and shape future investigations. However, the manual for preparation and publication of a review paper can often turn out to be an uphill battle. Welcome to this tutorial on how to quickly and efficiently generate a review paper using DORA, an advanced AI-driven tool designed to streamline the process of drafting academic papers and related documents. Let's say that we are interested in AI models that can predict functional properties of genomic sites based on DNA sequences. From the list of available templates, we will choose review paper, as we would like to prepare a publication on a specific field, focusing on the selected topic. In the input field that appeared, we should then enter the subject of the publication, which in this case will be convolutional neural networks used in genetics, to predict open chromatin regions based on DNA sequences. We then generate sections of the article by clicking on the button below. At this stage, it is possible to evaluate the generated table of contents. By default, 5 sections will be generated: abstract, introduction, main text, analysis, and conclusion. By clicking the plus icon on the left, you can enter custom data or additional subsections to customize the structure of the document. For now, let's leave the sections as they are, as the composition of the paper looks great. We will now generate the document overview by clicking on the button below. Based on the title of the article, DORA will prepare a content plan for the document. You can now review the recommended content outline. The option to edit the content is available on the left side using the plus icon. The proposed main text looks good to me, so I will proceed to generate the final document by clicking on the green button below. DORA agents are now creating the review paper. It might take up to 20 minutes. In the meantime, you can explore other features of DORA. You can also close the browser and take a short break, as you will be notified by email when the document is ready. The review paper is now prepared. DORA has also generated a bibliography based on the citations in the text. When you click on a selected citation in the text, a text evidence box will be displayed on the right side with an excerpt from the original publication from which the citation came. You can look at the details of the publication by clicking on its PMID at the end of the text evidence box. If you would like to add your custom text with a citation, click on the Add Citation button on the left side of the screen and search for the publication you are interested in through its keywords. If the publication is in our database, you can add that citation to the document. The newly cited article will be automatically added to the bibliography as well.
Hello colleagues, my name is Alex Pivorsky. I am a senior bioinformatician at Insilico Medicine, and today I would like to share with you one of the case studies we recently performed with our collaborators from the University of Copenhagen. There is no doubt that the radiotherapy is a key cancer treatment, but its effectiveness varies across cancer types and patients, which poses challenges for optimizing strategies and improving survival. Here we collected radiotherapy phenotype and expression data from 32 TCGA cancer datasets and performed overall survival analysis for 32 cancer types. Our findings show that radiotherapy improves survival outcome in certain cancer types, such as glioblastoma multiforme or GBM, while worsening outcome in others, such as low-grade gliomas or LGG. GBM and LGG are particularly interesting to study together because GBM often originates from a pre-existing LGG, representing a progression from a lower grade to a higher grade malignancy. This progression is associated with significant changes in gene expression profiles, which may underline the differences in radiotherapy outcome observed between these two cancer types. So, we next explored molecular mechanisms behind these differences, finding that pathways related to programmed cell death, DNA repair, telomere maintenance, chromosome consolidation, antiviral response, and interferon signaling differ between GBM and LGG radiotherapy treated patients. The identification of key pathways involved in radiotherapy resistance and sensitivity offers potential targets for future therapeutic strategies. With these findings, we decide to use DORA to streamline the process of drafting a scientific paper. So let me show you how to utilize DORA with already pre-existing results that you are willing to put in your paper. So let's start. First, we will need to select a template that is going to be used for a draft generation. Then we're going to add additional information as an input. And finally, we're going to take a look at the generated text by DORA. So to start with, we need to push on the plus button and select the most suitable template for a paper draft generation. Since we already described the results that we're going to put in our paper, the research article template, which denotes explore your data closely and integrate it with existing knowledge, is our choice. So, as an article topic, we will put variability in radiotherapy outcome across cancer types, a comprehensive study of glioblastoma multiforme and low-grade glioma, and then we're going to generate sections of our draft paper. So here is the proposed by DORA structure of the draft, and as you can see, our draft paper will include abstract, introduction, materials and methods, results, discussion, and conclusion sections. So since we already have results section, we can manually edit here by clicking on plus near results section button and custom data. So here we are going to put already existing text that describes the results that we got from the experiments, and then we're going to put on the submit button. So now when we will put on the button to generate document overview by DORA, it will include these results of the generation of the draft. We're not going to generate draft of the paper on the fly, basically because it will take some time. Instead, let's take a look at the already generated file that was used as a draft in this case study. So here you can see a draft of the paper. All sections, including abstract, introduction, and other sections are already filled with the text. And on the right panel, you can see bibliography, which includes all the references that were cited in the generated draft. And for example, by clicking on the specific references, we can be transferred in the specific text evidence why this reference was put here. And if you want to find additional information about this article, you can click on the PubMed ID number, and you will be transferred in the publication preview page of the specific article that includes abstract of the paper and other relevant information of this paper. Besides that, if you want to find the specific references for a specific part of generated by DORA text, you can underline the text and click on the Find References button. And let's check what references were used to generate the specific sentences. And here we can find the relevant papers that can be used as the references here. So as you can see, DORA is extremely helpful for drafting a paper. You can edit documents, add citations right here, or you can download the document and work with it outside. And basically, in current case of study, we used this draft of the paper. We manually curated it, extended a bit, and submitted into medRxiv with the University of Copenhagen.
Hello and welcome to Insilico. Many of you got to enjoy, use, and love the many generative tools that Insilico has developed for target identification, biomarker development, personalized medicine, and drug discovery. One of those tools is Biology 42 PandaOmics. We wanted to go way beyond what is currently required by the regulators around the world, even in some of the harshest environments. It's a box that does not connect to the internet. It doesn't have Wi-Fi. I'm going to reveal it today. And as you can see, we have two hardware solutions. One is a large PandaOmics box that you can put on your desktop and also put on your server rack. And we have a smaller device that has pretty much the same capabilities. It's a little bit cheaper, and you would be able to only utilize it on your desktop. For PandaOmics server, you can plug it into your server rack and so remove the sides, and it natively plugs in. plug the Ethernet RJ45, and that's it. You're ready to go. It doesn't have Wi-Fi. It doesn't access the cloud. All you need to do is from time to time get the regular updates from our cloud in order to update your models. So Insilico has developed the Insilico Secure Application Platform, ISAP, that allows you for chip-level confidential computing. It has enough GPU to power large language models. It also has all the latest scientific databases, the latest machine learning algorithms, latest large language models, and it has open API. Via open API, we allow you to directly communicate and interact with PandaOmics AI models. You can build and customize your own scripts and also utilize PandaOmics data for many other applications. So you could interact with PandaOmics in any way you like. The device is already ready, so you can request more information about it from our team and start preparing for shipping. So we're shipping this year. Welcome PandaOmics Box, and please do collaborate and contact our business development. Thank you. In today's turbulent world, it's very important to think about data privacy, data security, way beyond what's required by the regulators. Today's regulatory norms may not be the regulatory norms of tomorrow because technology is a moving target. There will be no new threats related to the data privacy, data security, and to go beyond, we decided to develop a local solution that is completely abstracted from the internet, completely abstracted from the cloud, and allows you to perform very advanced target discovery, biomarker development, drug discovery, personalized medicine tasks in one place, on one box. And it contains some of the latest advances in large language modeling, in other forms of AI that allow you to pretty much go beyond what's possible within the current drug discovery toolkit. Much of the tools, many of the tools that we've developed within the PandaOmics universe and in clinical universe, so Biology 42 and Medicine 42, are integrated into this device. You don't need to send your data to the cloud, and you can do all the compute locally. We went even beyond that and introduced chip-level secure compute into this device, so you can be 100% sure that the data is immutable and private and stays in the box. It does not have Wi-Fi access. You plug in your local RJ45 into your intranet, and the data only goes into the box. You can update the models, you can update the data, you can add additional capabilities that you are developing yourself, but us as Insilico and our ecosystem do not have any access to your data, your models, your hardware. It's completely separated. You can put it in a bunker and it will perform all the latest tricks for target discovery, disease modeling, and personalized medicine. I think it's very, very important to think about data as not something that is harmful or could be used to harm the patient, but something that is super valuable. Your data is gold. And to ensure that your gold is secure, this is the ultimate vault that allows you to create finished products from this gold to ensure that it becomes even more valuable. And today what I see is that many hospitals, many academic institutions, and many biotechnology companies are sitting on their data afraid of putting it in the cloud because they might lose some value or breach some law, or just because they're afraid that this data might leak. This tool allows you to unlock the value of this data, do local compute, identify targets, create valuable therapeutic assets that might help patients around the world, that might be patented and sold by the hospital, exchanged with the pharmaceutical and biotechnology companies, and allows you to completely abstract from the internet. Again, it doesn't have Wi-Fi. So what we have done here is enabling hospitals and biotechnology companies and academic institutions to go above and beyond current regulatory norms, and we deliver enough compute to perform some of the most advanced drug discovery, target discovery, personalized medicine tasks. This box can run a fully automated lab. It has latest GPUs that are available within certain regions. So we need to ensure that this GPU does not breach any laws and export restrictions. We've thought about that, and to run many of those models that we have in the box, you actually don't need that much GPU compute. You can also do some fine-tuning, local programming, and hook it up to your own data servers that are hopefully also disconnected from the rest of the internet. So welcome PandaOmics, PandaOmics Box, that allows you to do target discovery and disease modeling on site. Thank you, and start pre-ordering.
Hi everyone, my name is Khadija, and it's my pleasure to introduce you to Precious 3 GPT, the latest and most advanced addition to Insilico Medicine's lineup of Precious models. It's the next generation of our Precious models and the first language model that can generate and interpret omics-level data for practical purposes, such as preclinical and clinical trial simulation. Precious 3 features a novel GPT architecture trained from scratch to solve the problems of biodata domain transfer. We succeeded in creating a language model that can natively operate with omics-level data thanks to its unique data representation strategy and the inclusion of text and knowledge graph dimensions with the modality mapper units. Precious 3 is trained on a collection of over 1 million epigenetic, transcription, and proteomic observations from public repositories. This enabled the model to learn how these different dimensions of molecular biology form a single entity or a process. Precious 3 is a uniquely multimodal, multi-tissue, multi-species, and multi-omics language models that we're going to improve by relying on the feedback from our in-house multi-agent system and the community. You can download the model for your own Insilico experiments at our Hugging Face repository. We prepared all the materials that you may need to work with the model locally, including tutorial notebooks and documentations. For example, this Python notebook will teach you how to generate novel potential geoprotectors. So first of all, you need to authorize with an endpoint via token. Once you have configured your connection, you're all set to send JSON formatted requests to a Precious 3 endpoint. As you write the query, fill in as many fields as it makes sense and leave the fields that you want Precious 3 to generate blank. You can look at all the valid entity names on our Hugging Face. Once your JSON prompt is ready, add execution parameters to your request and submit it to the endpoint. If everything goes right, you'll get back a dictionary with a done message. In another dictionary entries, you'll find the actual text prompts used by Precious 3 and the model's output. When executed in the meta to diff mode, the output is 2 lists of genes, one for over and one for under expressed genes. Now you can continue working with Precious 3 GPT and resubmit the first output, changing the instructions or some other parts of the prompt. Alternatively, you can use one of the advanced execution models that automatically chain multiple Precious 3 requests and provide you with the final result, such as the compounds targeting a disease. With this notebook as a template, you may launch your own digital experiment and study aging, cross-species molecular biology, and hold frame using similar approach. Now let's move forward. Using a similar approach, we shortlisted 22 compounds and tested them in vitro in our robotics lab. Impressively, 8 of these compounds showed xenomorphic or xenolytic action, yielding a 36% hit rate. We are also developing a custom library to compare different LLMs in such biomedical tasks, which is basically an autonomous multi-agent system that can fetch all necessary data from the web, format it, select and execute statistical tests. On the screen, you may see an example of one such agent at work. The agent is instructed to use model embeddings to train an aging clock. Agent's thought process is on full display here. You can see it reason as it identifies the configuration of the model and as it modifies an existing script to work with a new configuration. Combining this agent with other agents responsible for model training, validation, and reporting, my colleagues and I can offload many routine problems and focus on more creative and thoughtful tasks. If you are not fluent in Python, you still can try out Precious 3 at our community Discord server. The Precious 3 Discord does not offer the full functionality of the model, but it is free to use and requires no setup. To start emulating biomedical experiments, you need to type !start in a specific channel. And in a moment, you will see a new private thread where you may choose from a number of workflows to reproduce with Precious 3. First, let's replicate a case control observational experiment and see what genes are up or down regulated in a condition. Click on the corresponding button and proceed to define the setting of your digital experiment in more detail. Surely you need to define the subject species. Let's go with human this time. Next, specify whether you're interested in exploring epigenetic or expression differences between the study cohorts. I want Precious to show me differentially expressed genes. We're almost done. I want to see how a condition affects the lung tissue. And finally, define the condition characterizing the case group. This will be lung fibrosis. You get a chance to review your request before submission. Everything seems right, so let's proceed. Precious 3 will now return a list of human gene symbols that are up and down regulated in human lung fibrosis. You may use these with external pathway enrichment tools or copy them for downstream analysis. Now let's go back to the main menu and try something completely different. This time we'll ask Precious 3 to run an in vitro senescence model and propose compounds to counteract it. For this type of experiment, you'll only need to select two options: the cell line in your digital experiment and the omics domain in which you want to erase the effects of cellular senescence. Proceed and check the results. And that's it. You have a list of 5 compounds that are free to explore on Chembol or consider for your real-life experiments. If you want to try Precious 3 GPT yourself, just follow the links provided by our community manager. Altogether, we're quite proud of Precious 3 and encourage you to try Precious 3 for yourself. Our studies have shown that despite a low number of parameters, roughly 900 million, Precious 3 shows state-of-the-art performance in multiple biomedical tasks, even when compared to the text-based models with over 8 billion or 70 billion parameters. We see this as an indication that our unique approaches for omics research, representation, and combining cross-domain sources has not yet reached its full potential. We also hope that Precious 3 will incentivize AI and aging researchers to adopt this new multimodal methodology and achieve even greater heights. Our team for sure will continue working to improve all Precious models and combine them into a single system that will enable fast-paced, affordable, and innovative biomedical research and drug design.
Hi everyone, Daniel here. I'm a Senior Director of Technology at Insilico, responsible for our product management, AI research and software development. Today I'll give you a brief update on 3 of our platforms, but I also invite you to participate in our Pharma.AI Day this November, where we'll introduce major updates to all of our platforms and tools. Pharma.AI spans 3 pillars of drug discovery: biology, chemistry, and medicine. Together, they help you discover and validate biological targets, create small molecules and biologics, and analyze clinical trials. At every step, we help you advance your drug discovery program and make informed decisions based on reliable models and data. Let's start with PandaOmics. PandaOmics is our target identification platform. It helps you navigate vast amounts of curated data, including omics, publications, patents, grants, clinical trials, and more. Our AI models help you find connections between genes, diseases, and drugs. In the recent updates, we've enhanced the way users interact with the platform and enrich the data. We've improved gene-disease association reports, streamlined meta-analysis setup, and introduced genetic evidence for gene-disease associations. These updates are already available to all of our users, so let's see them in action. When you investigate an association between a gene and a disease, you want to extract the most relevant information from the vast amounts of available data. PandaOmics will help you with this by writing a comprehensive report, summarizing relevant papers, analyzing available omics data, investigating mechanism of action, and proposing a path to market. Here, we want to analyze the association between ALS and IL-6R. To generate a report, I click Generate. The report will be available in a few minutes here or in the Data Manager, where all of your data is stored. Currently, multiple LLM agents analyze the data, summarize it, and draft the report. With the last update, LLM reports became more powerful and accurate. The report cites all the relevant papers, visualizes the differential expression heat maps, and knowledge graph, as well as providing path-to-market information. As you saw with the LLM reports, streamlining your research is our goal. The next step is analyzing raw data in more details to test your hypothesis. You do this by running an omics data meta-analysis, comparing different samples across multiple gene expression series. Selecting the right data is crucial, and our new AI-based experiment designer will quickly find relevant datasets. From there, you can review the details and narrow down the list. Now, I can launch the calculations. We can review the results to find over- and underexpressed genes, as well as affected pathways. We can also analyze how different comparisons align with each other. Is a gene under or overexpressed in all comparisons or only in some? If the results are consistent, there is a strong evidence that the gene is involved in a disease of interest. Besides finding a target for a disease, you can also do the opposite. Find disease caused by a given target. In the new version of PandaOmics, it is easy to analyze specific gene variants or mutations implicated in a given disease. Open a gene page and go to the Genetics tab. Here, you can see the diseases known to be associated with this gene. You can also dig deep into the data and analyze specific pathogenic variants. With this information, you can prioritize the diseases and confirm the gene-disease association. Next up is Generative Biologics. Generative Biologics is an advanced AI-powered platform for designing and optimizing different types of biologics. With the latest updates, we can now generate alpha helical peptides, nanobodies, and antibodies tailored for specific targets. We have also incorporated an AI-based affinity prediction model to further improve the potency of generated structures. Generative Biologics allows our customers to design structures with a desired functional response. You can specify the exact epitope where you want an antibody to bind. The most important and exciting thing about the platform is that it has been experimentally validated and demonstrated excellent performance in the design of novel peptide binders. We chose a well-known GLP-1 receptor for our de novo design experiment. We have also removed all proteins similar to GLP-1 and GLP-1R from our training sets for the fairness of the experiment. The platform identified the binding site and produced over 5,000 peptides in 72 hours. We selected the top 20 of them based on the predicted affinity score and tested them in the wet lab. Out of these 20 peptides, 12 were active and 3 of them showed a single-digit nanomolar activity. We are proud of these results as they build confidence in our platform. Let's now take a look at the platform itself. The first workflow is peptide design. We press the Create Experiment button and select the generation of alpha helices. We now see the experiment setup screen, and the main thing here is to upload the target PTB. Our AI model finds the appropriate binding site, and then we should specify the desired peptide length from 8 to 60 amino acids. And also the time to run option controls how long the peptides will be generated for. After that, we press run and launch the experiment. In several hours, you will receive the results. They are presented as a table, and each row contains one generated peptide. Clicking on the row opens the molecule card showing detailed information. We can modify the visualization slightly to obtain a better view. We can also extend the list of calculated parameters that helps us select the most promising molecules. All the results can be exported to a local machine for further analysis if needed. The second workflow I wanted to demonstrate is the antibody generation workflow. Here the logic is very similar to that of peptides. We create a new experiment and select antibody generation. We also need to upload the PDB file in the setup experiment window. After uploading the protein, you can mark the needed amino acids using the table on the left. When you select amino acids, they are marked in green in the visualization window. To ensure that you selected the correct amino acids, you can change the representation for a more convenient view. When the epitope is selected, it is time to set the time to run and launch the experiment. The results look very similar to those of peptides. However, the calculated parameters differ. If we go to the molecule card, we can see how the antibody interacts with the receptor, and again, we can modify the view to look at the contacts. The list of parameters can be further extended, and we can select all of or several antibodies to download to your local machine and analyze them further. Let's now switch to Chemistry 42. Chemistry 42 is a comprehensive small molecules drug discovery platform. It combines the flexibility of generative AI and the accuracy of physics-based methods to create optimal molecules. There are 4 apps in Chemistry 42. With generative chemistry, you can create new small molecules or rank existing ones to prioritize active molecules with desirable ADME properties. When you have a short list of compounds, you want to prioritize or filter them down further, you can use alchemistry, a physics-based algorithm to accurately estimate relative binding free energy. We also have 2 predictive model tools, ADME profiling and golden cubes. ADME models estimate physiochemical properties, absorption, distribution, metabolism, excretion, and toxicity of your molecules. You can use them as a standalone tool or include these properties in the design within generative chemistry to optimize ADME properties of your lead compounds. Finally, if you work with kinases, you know that kinase selectivity is a big challenge. Most active molecules will affect not only your target protein but also many off-targets. We created golden cubes and kinase activity and selectivity profiling tool to facilitate the development of kinase inhibitors. Over the past several months, we have significantly improved the platform's efficiency and user experience. First, we have significantly improved our alchemistry platform, making it more accurate and efficient. We are also democratizing small molecules design. Academic institutions and small biotech companies can now purchase and manage their subscription directly in Chemistry 42. We have also introduced interactive walkthroughs and demos, making it easy to get started and learn the best practices. I'm excited to share recent updates on alchemistry, our physics-based workflow designed for accurate binding free energy estimations. Since its introduction, we've significantly expanded alchemistry applicability. It now supports calculations for protein structures with non-standard residues, complexes containing cations, and organic cofactors, also membrane protein systems. In this demonstration, we will conduct simulations for the membrane-embedded G-protein coupled receptor mGlu5. To enable a membrane system, navigate to the Configuration tab and select the appropriate checkbox. Returning to the Input tab, let's begin by uploading ligands. Simply drag and drop your file into the interface and click Next. Then select the columns with IDs and experimental data. Be sure to choose the correct units of measurement for your data from the drop-down list. Now let's upload a protein structure. For membrane-embedded receptor simulations, you need to upload a pre-prepared system. In the visualization box, you can manually inspect the entire system and the uploaded ligands. Now you can select the compounds to include in the calculations. For this demonstration, I will choose 5 compounds and perform simulations in a zero-based mode. You can adjust the number of tokens to speed up the calculations and then click Run. The Results tab displays ligand structures and binding energy estimates, which are available for download in SDF or CSV formats. We've also incorporated a new tab called Insights. This tab provides a detailed overview of all perturbations in your experiment, making it a pretty convenient tool for monitoring the experiment status at any time. We highly recommend using the Insights tab if you need comprehensive details about your experiments. Academia and small biotech can now purchase and manage Chemistry 42 subscriptions directly on the platform. Once you access it, you can click an Activate button, or once you attempt to run an experiment, it will also prompt you to purchase a subscription. You can select your subscription tier and pay the license fee with a credit card. I won't show you this part. After purchasing, you can immediately run the experiments, and you can update your subscription at any time in the organization settings. Finally, when you first log into the platform, you will see a set of training videos introducing to the platform and explaining the best practices. For every experiment type, the platform will show you an interactive walkthrough, helping you set up your first experiments. You can also ask our Chemistry 42 GPT about the platform.